And both our previous speakers have already referenced his work today. And the final talk of our session is given by William Greenleaf from Stanford University, who will provide us with some insights into understanding the physical genome in health and disease. So over to you, William.
Okay, let's see if I can present. Well, thank you so much. I'm stuck in California and it's real early for me. So I wish I would have gotten up a little bit early. I didn't quite see all of the talks, but I'm very heartened to hear that other people are using display techniques on the sequencing instrument. So I decided to sort of focus on that today, but I wanted to give you a quick background of the other things that are happening in the lab, where my lab is generally excited about exploring the physical genome, which I define as understanding first how the genome is folded and how this affects gene regulation. And we've developed methods for probing chromatin accessibility in vivo or in cells, methods like ATAC-seq and also, single cell methods for probing chromatin accessibility, and apply these to things like understanding human differentiation patterns and also cancer. We've also developed other single molecule methods for understanding chromatin accessibility that use long reads, and also methods that use ionizing radiation actually to make correlated spatial breaks in chromatin and understand structure. But I'm not going to talk about any of this today. Instead, I'll be talking about the sort of other side of the lab that's excited about understanding sequence structure function relationships, and this, oh wow, okay, wow, there's a lot of delay. This involves using sequencers to, for example, display RNA, to understand RNA binding protein interactions, RNA tertiary structure, using these high throughput methods for display to allow people, for example, on the internet to design RNAs and then test them in real life. Understand CRISPR affinities, understand the sequence specificities of AGO2 binding, and also protein display. I think some of that has already been discussed. So I'm going to sort of blast through my introduction of the system. But here's a little bit of motivation, my spin, I guess, on the motivation. Everybody's seen this figure before, and unfortunately, it ends in 2012. I think it would be sort of down here, maybe today. But obviously there's a revolution in sequencing technology. I asked I think this is faster improvement of technology than any technology ever, other than like fire, maybe, I don't know. So this is really causing a revolution in biology, and maybe we can sort of use this technology to try and solve this functional genomics bottleneck. How can we relate, for example, a massive catalog of sequence changes to functional changes, and really larger, this classical question in molecular biophysics, how does changes in DNA sequence or the encoding polymer sequence affect changes, affect the structure and function of biomolecules? And if we really understood this, of course, we could call out a function, and then I could tell you what sequence to use to carry out that function. And I think we're pretty far away from that. But my claim is that what we really need to do is make billions of measurements in order to build an understanding of how these sequence structure function relationships work. So we might actually build predictive models for design. So there's lots of scales of biochemical functional assays. Obviously, you can do things in tubes and 96 well plates, and microfluidics and microarrays, and of course the ultimate, which we've already seen, single molecules. But this Illumina sequencer sits in a really juicy sort of area in this continuum where you're not trying to image single molecules, which, as you can see, are hard to image, and you are also able to pack a billion features, a billion different experiments effectively onto something the size of that you can hold in your hand. Okay, so we've talked about all this before, apparently. So we've got a lot of little clusters on a sequencing chip. What you can do is have a RNA polymerase initiation site, for example, initiate the RNA polymerase, have it make RNA that it then gets stuck at the end of this a construct, and now you've displayed your RNA, and you can look at RNA binding or RNA-RNA interactions and things like that. Okay. We actually have been pirating old GA2s to take the MiSeq chip and image them, and we're kind of moving, I think, more toward purpose-built instruments too, until Illumina allows us to buy one, apparently. So that would be wonderful. Okay, so using old technology, you can clone these things for less than $10,000 a piece. Okay, so let's use this to answer some questions. And this is a collaboration with the Hirschlag and Doss labs here at Stanford. But as we know, there's really reasonable models to take, for example, RNA primary sequence to secondary structure and understand the thermodynamics of that folding. But actually going from to tertiary structure is still a hard problem. Understanding how these different sort of secondary structure elements interact with one another, and what the ensemble of conformations is of a tertiary structure, given any RNA sequence, for example, is really unsolved. But there's hope, right? There's conformational ensembles, there's stability conferred by distal interactions. But there's hope because if you look at folded RNAs, you can pick out these sort of functional elements that seem to be repeated in lots of different RNAs. So helices, of course, different kinds of junctions, different kinds of tertiary contacts. And you can see these over and over again if you look at the crystal structures of RNA. So maybe if we can characterize exactly how these building blocks of RNA behave, we might be able to come up with a predictive model for how they might fold in solution for unseen molecules. Even really simple questions like, for example, if I have different Watson-Crick base pairs in a helix, are those interchangeable in a structure, or do they actually cause differences? I think a lot of people would say, oh, those are all the same. And for example, also, what about two base bulges? Is an AA bulge the same as a UU bulge? Does it bring about the same sort of conformation of three-dimensional or six-dimensional spaces, conformations of the ends of this piece of RNA? So what we wanted to do is try and build move towards a predictive model for tertiary structure. So what elements, how does RNA behave modularly? Can we break down RNAs into pieces that sort of behave the same way? And can we develop simplifying rules for this behavior with building blocks? So again, we can think about the fundamental building blocks as helices and junctions would have bulges or mismatches, bulges in multiple directions, mismatches like this. Can you guys see my pointer? Maybe not. Oh, you can. Great. Okay, so we tested this in what I like to call that sort of hydrogen atom of RNA tertiary interactions, and this is the tecto-RNA system. So this is a heterodimer of RNAs. There's one here that has a receptor and then a helix and then another sort of tertiary interaction. This is actually on the order of a nanomolar affinity. And what we can do is simplify this to this. We can think about putting one of these on a chip and flowing in the other one, and then asking if I change sequence to the chip piece through this huge library of different sequence elements, how am I affecting the affinity of this probe or this flow piece? And that, of course, has to do with what the conformational ensembles are and if they are overlapping in ways where they can actually make this tertiary interaction. And so we can sort of insert different length helices with different RNA, with different sequence contexts. We can add different bases and mismatches, and we can also even add different tertiary contexts. Okay, so how does this work? Again, we've talked about this. You make polymerase, you make RNA, you add your flow piece, and you measure the fluorescence. And again, this has been covered, so that's great. I don't have to cover it again. So how does this look? Well, we, again, every cluster is a different RNA. We add more and more RNA, different tecto-RNAs on the chip bind at different concentrations of the flow piece, and we get different binding curves. And you can see very reasonably, as we change the length of the flow piece, we get really tight binding and then bad binding, because there's sort of sweet spot for binding when the length of this flow piece is just the right length to make this tertiary contact with the RNA on the chip. And the nice thing about this is it's incredibly reproducible. We can get really sensitive and reproducible across multiple log scale of KD. Well, this is about 4 logs, and it's quite reproducible across experiments. So our relative binding energies, I think, are incredibly good. Okay, so first question: how much does sequence variation matter for this sort of affinity? If I have a constant length and I just change the Watson-Crick base pairs, how much change in the affinity between this tertiary interaction do I get? And surprisingly, we see actually a ton of variation. So this is the average error per variant, and we see, 2 or 2.5 kcals per mole worth of energy, which my collaborators were very surprised at. And it's really nice to be able to sort of observe this directly on the chip in large numbers. And I think the question is, can we predict anything about this? And my collaborator, Riju Das and Joe Yesselman, had this really nice sort of empirical model for building up ensembles of different RNA structures. And so what you can do is just grab base pair steps from crystal structures and then sort of add them together into many different ensembles of structures. And when the ensemble of structures has basically is within a distance that you could make this tertiary contact, you're going to score it bound. And when it's not, you score it unbound. And that's a really simple sort of partition function that you can use to get a delta G. And lo and behold, this relatively simple empirical model based on empirical observations from crystal structures predicts with no free parameters quite well the observed delta G with maybe explaining 70% of this variance. So that's really nice. And again, the differences between these affinities are non-trivial, but if you stare at this sequence, I don't think any human could realize that those have very different conformations in three-dimensional space. And if we just ask about the relative contributions of every base, here is what we observed, and here's what is predicted by these models. And so it actually is recapitulating what we see really well. But can we get a better physical understanding of what's going on there? So what we can do is take all of these different models of the end-to-end conformations, the ensemble of conformations of all these different helices, and do principal component analysis on them and say, what are the general ways that when you change sequence, how do you vary in 6-dimensional space? And so there's two major ways of varying, and some of them sort of vary in X and Z and 1 and Y. And you can see actually, but the principal components that vary with the predicted delta G, it's almost entirely this principal component too, which really is this Y dimension of variation. So as you change your sort of distance in Y, you become more or less likely to bind to that tertiary interaction. Okay. So that's great. Base pair steps are modular, but sequence actually substantially influences helix behavior in terms of the binding, and so now we have a predictive set of rules. But can we extend this to other things? Well, I want to introduce this idea of a thermodynamic fingerprint, where, for example, we can have on the flow cell a length 9 helix, and we can flow in a length 8 helix. We can flow a length 9 helix into solution, and on the chip we can have length 8, 9, or all the way to 12. And you can imagine that's a different conformation of states that are capable of binding at different positions in X and Z, in Y and Z in this case. As you add bases, you sort of twist and move away. And there's different overlaps with this 9 base pair helix, but we can also change that helix that we're flowing in and ask how that affects where you like to bind by varying both the chip and the flow piece lengths. And what you get then is, as you look at the affinities, is this sort of thermodynamic fingerprint, we call it. Okay, so what does that look like? Well, if you look at all, this is a 1-by-1 mismatch, this is a 2-by-2 mismatch, this is a bulge of one base, and if you cluster all of these different types of bulges, you can see that the first thing that clusters is the number of mismatches. You basically cluster by the number of mispaired bases. That sets your general behavior with respect to binding. But if you look, this is again averages across the class, but if you look, there's actually a ton of variation, especially for example 3-by-3 within this class. And so maybe we can understand even more about the details of these bindings. So just as an example, because there's tons and tons to do here, obviously, let's just look at mismatches. If you look at a mismatch at different positions and you build these thermodynamic fingerprints and cluster the different behaviors of different mismatches, you get sort of 7 different clusters. And in reality, and what that is if you look at the fraction of different mismatches in each of these sort of behavior classes with respect to the binding affinity to all these different partner molecules, you can see that the GG mismatches sort of form a cluster, CU and CUC mismatches form a cluster. And then actually a lot of these mismatches look a lot like Watson-Crick bases, actually. But so there's actually a lot of structure here. AA, GG, CU, CU, CC are distinct, and these other mismatches actually behave quite a lot like actually just base pairs, surprisingly. We can also probe bulges, for example. So in this case, we have a bulge in the 3' or 5' side in 2 different contexts. So with base pairs of different types. And we can do the same trick. We make lots of measurements. We cluster their behaviors using this thermodynamic fingerprint. And then we can look at the principal components. It turns out that the majority of the behavior can be, the majority of the variance in the affinities can be explained by differences actually in this flanking sequence, which was kind of surprising to me, that was a major determinant of the overall conformation, not the type or even the side of the bulge. The side of the bulge has a big effect. It's 20% of the variance. And then the actual base that is in the bulge is actually a relatively small effect size. So that doesn't have a huge perturbation. Okay. Really quickly, the energetic, so we can also use this sort of statistical model where we grab different conformations and predict affinities. But unfortunately, for a lot of these junctions, there's either no crystal structure or there's only one crystal structure to choose from. So we can't build an ensemble of states. And it turns out we predict much worse than these Watson-Crick motifs, as you can see here in this heat map. But we can actually solve that by bootstrapping some of our thermodynamic measurements. So it turns out there's lots of neighbors, there's lots of junctions that behave very similarly to you if you are an uncharacterized junction. And some of those junctions might have crystal structures. So I can take all of the junctions that behave similarly in this sort of thermodynamic fingerprint space and add them all together and make an ensemble of different possible conformations and use that to draw from and build an ensemble of overall states and then score that to try and build affinity. So here, for example, is an ensemble of one kind of junction, another. Here's an ensemble of junctions that all have similar thermodynamic behavior, and in this case often have a single base bulge sticking out. And so if we do this sort of bootstrapping, grabbing our neighbors in thermodynamic space and using that structure to make predictions about how I should behave, we can do much, much better in terms of our RSME error. So we can kind of understand structural characteristics of uncharacterized junctions using this method. Okay, now I want to change gears a little. Oops. Oh my gosh, I'm sorry. Trying to find the time. Okay, I want to change gears a little bit here because I'm really low on time, but here's another example of the platform to try and understand nucleic acid programmed protein affinities. And in this case, we collaborated with Phil Zamor to try and understand Argonaut-2, the core of the RNA-induced silencing complex RISC. So a lot of people try to understand how this thing binds and how it might knock down RNAs. And actually, a lot of the best microRNA prediction programs it's hard. It's a hard problem. And our hypothesis was maybe adding quantitative biophysical measurements of both binding and cleavage might improve our ability to model, in this case, cleavage directly by microRNAs. So again, it's the same deal. We make a tethered RNA. We add labeled AGO2 loaded with a guide, and we measure affinities on rates and off rates. And again, we make a big library testing all the hypotheses we want. I like to sometimes say, like, you can do 5 PhDs worth of work in one experiment because, there's 1,000 experiments for every hypothesis you want to test for 100 hypotheses. Okay, so again, we can measure on rates, and here's just some examples of this. And here's the classic plot where we can get the K-on. And so what does this look like? Here is a matrix of all double mutations of the target sequence. And you can see in this case that the on rates are substantially affected by mutating things like seed regions, which was to be expected. But this is just sort of an example of the kinds of data we can generate. We can also measure equilibrium binding curves. So again, this isn't on rates. This is waiting till we come to equilibrium and then approximating what we think the equilibrium constant is. And this is actually in picomolar. And again, we see mutate. This is again the double mutant map here, and we can also do this for example for triple mutations and ask about the sensitivity of mutations. The one thing that we also see is if we add insertions at specific places, sometimes the Argonaut doesn't actually care about those insertions, which really has implications with respect to how you might design algorithms for understanding where these proteins bind. We also sort of adapted this cleave and seek experiment to try and understand cleavage. We didn't actually do cleavage on the chip. We did this in solution, where we just make a bunch of RNA and then let AGO punch it, cut it, and then go on to PCR. And again, that's what these data look like. This is, again, like traditional sequencing. But the interesting thing that we see here is actually sometimes if you add mismatches, you can get faster cleavage than the fully complementary RNA target, which was actually very unexpected. And it has actually interesting applications and implications toward therapeutic microRNAs, because actually adding some mismatches at specific locations, double mismatches, actually improves your ability to cleave. And we think that's because of eliminating some of the steric hindrance to get into the cleavage state. Okay, but overall now we have kinetic measurements for on rates and equilibrium constants and cleavage rates. And so we can build a relatively simple model with only a few free parameters. And we actually did in vivo measurements of cleavage on a library. And I just wanted to show that you can actually do fairly well with your biophysical measurements, predicting how well things cleave in vivo. And there's a lot of caveats here, obviously, going in and trying to predict things in vivo where lots of other things are happening gets complicated. But we were pretty happy with this agreement for cleavage rates. Okay. And shoot, how much? I have no time left, right? That's cool. I just want to say one thing really quickly about a really fun collaboration we've been doing with Riju Das called EteRNA, which is an online game where you can design RNAs that are meant to switch between states, for example, upon the addition of a small molecule. And then you can submit your answers. We can synthesize those riboswitches, characterize them, see if they actually worked, and then return that information back. So here is sort of what this looks like. Here's an FMN riboswitch where you're supposed to actually fold into an MS2 hairpin here. This is bound by MS2 when FMN is around, and you're supposed to have a structure without FMN that doesn't have this MS2. So when you add FMN, you light up basically here. And so just a quick example of how this game is played. Here's the RNA structure. Those yellow guys are I think A's, yes. And now you can go in and change the sequence of each of these bases until you get the right conformation in the bound versus unbound state. And so people go in and change all of the bases, and they have interesting strategies. Okay. And then we can, again, we know how to do this. We assay them, and we can see, for example, here is a riboswitch where when FMN is present versus not, we get a huge shift in the binding affinity of the MS2, which is what we were aiming to design. Okay, one more quick thing. We can also do multi-input switches. So we tasked people with generating, in this case, an XOR gate. And you can see A, B here. Sorry, yes, XOR, yes. Not A, not B is off, and A and B is on, and you can see really nice behavior there. Okay, I am out of time, but of course, the most important thing is all the people who actually did this. My collaborators, Dan Hirschlag, Riju Namita, and Phil Zamore, and then all the other people in the lab who actually did this: Sarah Denny, Winston Becker, Ben Oberreynolds, and Johan Andreasen. Okay, thank you for listening to me.
Thank you, William, for that interesting talk. Don't think we've got any questions in the Q&A at the moment. Maybe I can start with one. So I wonder, can you adapt these methods to study small molecule induced changes in RNA conformation?
I think that the answer has to be yes. We haven't specifically, we've thought about doing that actually with certain interesting, potentially interesting systems, but all you have to just think about what is your fluorescent readout, right? Yep. And for example, we've also looked at aptamers where when you bind us, actually we've done this. We have aptamers where when you bind a small molecule, you sort of zipper up a hairpin and then get quenching, for example. So you can look at RNA or DNA small molecule interactions through this sort of stabilization of a quenched conformation. So yes, I have a demonstration proof for that one.
Okay, and I guess a follow-up question is that, you mentioned that, being able to take a function and design a sequence to achieve that function, we're a way off that. But are we any closer in terms of, being able to design small molecules that modulate a structure to give you a change in function?
No, I think we're still a long way from that, but my hope is that these extremely high throughput methods that provide quantitative data sets are excellent fodder for either neural network type models, but eventually I think physical like models that will have that use physics simulation type algorithms to predict what's actually bound and what's going to bind, what's not going to bind, dock and or that kind of stuff. So my hope is that with enough constraints, we will get to the point where we can build those sort of meta models that can make those sorts of predictions.
Yeah, that'd be good.
You're asking specifically about small molecules, which is sort of like an even harder problem, I would argue, but yes.
Absolutely. I work in the small molecule area and, it is difficult, but yeah.
No, I definitely, I do think that small molecule binding to like aptamers, I think that we have huge amounts of data on that. And I think that processing it to get to physical insights is now sort of one of the bottlenecks.
Yeah, that's great. Do we have any other? It doesn't look like we've got any other questions.
No, I think the reason for that is I posted the website for the RNA game in the chat. So probably
everybody's off playing the game. Yeah,
everyone is designing their own RNA.
Yeah. Oh, great. Well, that'll be great. Hopefully we can use some of those.
Excellent. I think if there are no further questions, then it just remains for me to thank William again and to thank all the other speakers in this session. Thank you very much, everybody.